Okay, welcome back, everybody. Thank you for being here again on this special day. I hope you all had a good lunch, and we are going to have another couple of great panels, and then a conclusion, and then a reception, so we can all hang out and have some fun. And since I know many of you are wondering, yes, I did change outfits in between the morning and the afternoon. I was wearing my fish shirt in honor of the Vertebrate panel during the morning, and I'm now wearing my butterfly shirt in honor of the Invertebrate panel this afternoon. So I hope that gives you a sense of my dedication to this topic. Although actually, my dedication is nothing in comparison to that of Sophia Fogel. I said I would thank her multiple times today. So Sophia, where are you? Are you here right now? Yep, there she is. There she is. To give you a sense of her dedication to this topic, when she was booking the room for this event, when she was interacting with campus offices booking the room, she was doing that from a tattoo parlor in Berlin where she was getting a tattoo on her arm of a spotted lanternfly. And that is not her first invertebrate tattoo. So that definitely has me beat. Okay, so we are now, and that is the perfect segue into going into the invertebrate session. So I'm going to kick it to my colleague, Jonathan Birch, to introduce the invertebrate session. Jonathan.
Thanks very much, Jeff. I don't have taxonomically themed shirts, I'm afraid. I am still dedicated to the topic, though. You just have to take it on trust in my case. We had a brilliant morning covering a lot of evidence very quickly in relation to vertebrates. I'm going to keep up the momentum this afternoon by now turning to invertebrates and presenting and discussing a lot of evidence again. First of all, some good news, which is that we're already up to 100 signatories on our declaration. Thank you. Provisionally, we will be individually verifying all the signatures, but provisionally we're up to 100, and we'd love to get more. We'd love this to be running well into the hundreds to show people what a substantial research community this now is. So, to turn to invertebrates, I will introduce the speakers one by one, and we'll be starting with Alex Schnell. Alex Schnell, very valued collaborator of mine over the years, and especially well known for her work on cephalopod mollusks, especially cuttlefish, and also corvids. Alex is a research fellow at the University of Cambridge, where she's worked with Nikki Clayton, and a National Geographic explorer. Her professional career has been directed towards furthering our understanding of the remarkable behaviors of non-human animals. Her research interests center around complex learning and memory mechanisms in animals and how these abilities have evolved across diverse taxa, specifically the cephalopods and the corvids. She's motivated to inspire compassion by working to deepen our empathy and understanding of other minds. Let's welcome Alex.
Thank you. I need a little stool. Exciting times. I'm so pleased to be joining you. And let's see, where's my talk? There you go. So today I'll be providing evidence of behavioural markers that suggest the existence of consciousness in cephalopod mollusks. Now, cephalopod mollusks include the shelled mollusks, such as nautilus, and also include 3 different groups of soft-bodied cephalopods: the squid, the cuttlefish, and the octopus. Oops. So the most widely accepted behavioural markers of consciousness are often investigated within the context of whether an animal can feel pain. And these markers suggest the animal is not simply having a reflexive response to pain, rather they're having a subjective pain experience. So the markers include, as we've already discussed, but I'll go through them quickly, motivational trade-offs in which the animal weighs opportunities for rewards against the risks of injury, flexible self-protection where the animal guards and grooms wound sites, and these behaviours suggest that an individual has some sort of internal representation of the injury. Sophisticated associative learning, where the animal learns to associate certain stimuli with unpleasant outcomes. And the type of learning I'm talking about usually involves decision-making and memory. And finally, analgesia preference, where the animal shows that they value pain relief when they're injured. Now, I haven't put references up on my slide. That's because the evidence that I'm showing today for these particular behavioural markers are at about, well, there's about 170 references, but I do have them if anyone's interested. So currently there's no direct evidence of motivational trade-offs in cephalopods, and this is not because there's evidence of absence, rather there are no studies that directly test for this or rule out alternative explanations. So there is, however, some incomplete or indirect evidence in octopus, cuttlefish and squid. Now, for instance, octopuses presented with hermit crabs, they're a common prey item, learn to avoid the hermit crabs with stinging anemones on their shells. They abandon hunting opportunities that repeatedly lead to stings, and they'll move away from hermit crabs with anemones. However, this study did not directly test for a motivational trade-off. To do this, the quality of the reward, so the hermit crab, would need to be varied to investigate whether octopuses would incur higher risks of stinging to access higher quality rewards. Cuttlefish exposed to infrasonic pulses, which mimics predatory attacks, they abandon opportunities to hunt and instead exhibit defensive behaviour. And what's interesting is that hunger affects their responses. So food-deprived cuttlefish have a higher escape threshold. Now, one explanation for this hunger dependence is a motivational trade-off, whereby the value of the food opportunity to the animal was weighed against the disvalue of threat exposure. However, an alternative explanation is that hunger simply inhibits that threat detection. Squid display changes in behaviour after injury. So they often use crypsis, a defensive behaviour commonly seen in cephalopods, to avoid detection, and they'll escape earlier when they're exposed to threat. Injured squid are also more likely to school in the centre of the group when a visual predator is present. One explanation for these patterns is that injured squid are aware of their injuries. They attach greater value to the need to escape and reduce predation risk relative to their other needs. And an alternative explanation is that visual and tactile receptors are sensitised, and that's not evidence of a subjective experience. So there is direct evidence of flexible self-protection in octopus and cuttlefish. And there is some indirect evidence in squid. So for instance, in squid that have been injured, they do show defensive arm postures and widespread nociceptive sensitisation, but they don't show protective behaviour that's directed towards the wound. I would like to say, though, these studies in squid, the site of the wound was at the back of the left fin, so it was virtually impossible for them to reach it with their arms. So we need further research in squid. Now, in octopuses, injured individuals will often hold their wound sites in their beak. One study has found that sites injected with acetic acid will evoke stripping behaviour, so the individual will strip away skin around the wound using their beaks. Injured octopuses also stroke the wound sites with the tip of their arms, and they even wrap and guard their wound sites with their adjacent arms. A recent study on cuttlefish has demonstrated that individuals perform wound-directed grooming, where they'll stroke wound sites with their arms, similar to octopuses, and they've also known to place their arms around post-surgery sites as if to guard the area. Now, there's clear scientific consensus that octopuses and cuttlefish are readily capable of sophisticated associative learning, and the evidence is weaker in squid and nautilus. So octopuses have impressive learning abilities that go well beyond simple forms of classical conditioning. They associate reward or punishment with various stimuli, but they also show types of associative learning that are strongly linked to sentience, including reversal learning, spatial learning, God, this has a mind of its own, and learning across temporal gaps. Cuttlefish associate reward or punishment with various stimuli as well, and I think there's perhaps even more evidence of types of associative learning that are strongly linked to sentience, including instrumental conditioning, reversal learning, spatial learning, aversion learning, and learning across temporal gaps, both short and long. Squid associate punishment with various stimuli. So a recent study on bobtail squid showed that they can learn to avoid striking a prawn inside a glass tube, and this is a type of instrumental conditioning. However, the researchers of this study failed to exclude habituation as a potential driver of this pattern. Now, one study in Nautilus found tentative evidence of spatial learning. So specifically, the Nautilus were placed in a maze and they had to find how to exit the maze. And so the reason I've said that the evidence is tentative is because the Nautiluses showed a reduction in exploratory behaviour on repeated exposure to the same maze. So they would get into the maze and beeline it for the exit. So this could be a form of habituation. And there's direct evidence of octopuses showing a preference for a location in which they can experience pain relief. An important study by Robin Crook probed this question by using a condition place preference test. So in A, you can see the octopus in the apparatus, so on the left, and on the right, this image represents the diagram of the apparatus, showing that two different chambers are distinguishable by different patterns. Now, octopuses were first presented with a free choice between the two chambers to move about, and their initial preferences were noted. Then some of the subjects received a noxious stimulus, where an injection of acetic acid into the arm was administered, and they were placed in their preferred chamber to experience the effects of that pain. Later, some subjects received a local anaesthetic on the injury, and they were placed in the other chamber to experience the effects of pain relief. Now, the question that Crook posed is, would the octopuses switch their preferences? Would they develop a lasting aversion for the chamber where the effects of injury were experienced, and a lasting preference for the chamber where the effects of pain relief was experienced? And that's exactly what the octopuses showed. And in mammals, such as rats and mice, this preference shift for both the preference and aversion patterns is taken as evidence of pain experience. Now, cephalopods display other advanced cognitive abilities that have been linked to consciousness in humans, such as remembering the past. So octopuses appear to recall where they recently explored, and they avoid areas that they recently depleted of resources. They also appear to understand the chronological order of their personal experiences. In cuttlefish, they demonstrate capacities for episodic-like memory. Specifically, they remember what they ate, where they ate it, and how long ago. And cuttlefish can even retrieve the sensory source of a foraging memory, so they know whether they have seen a prey item or smelt it. Now, other cognitive abilities that are associated to consciousness in humans is the ability to plan for the future. So cuttlefish exhibit traits that suggest at least they have precursors of this ability. They demonstrate remarkable self-control, waiting up to 2.5 minutes for a better but delayed reward, which reflects an orientation towards the future. And cuttlefish also plan for imminent future events. So for instance, they adjust their foraging strategies based on learned patterns of prey availability. Specifically, they will eat less crab for lunch when they can predict that shrimp is on the dinner menu. Now, octopuses exhibit an active phase of sleep, and they show behavioural patterns that suggest they could be dreaming. So in this phase of sleep, octopuses display short, irregular and rapid changes on their skin, and they show muscle contractions. And these type of behaviours really resemble the way that they might respond to a prey item or a predator when they're awake. And in humans, this active phase of sleep is called REM, which is associated with dreaming and linked to consciousness. Other evidence points to glimmers of environmental and social awareness. So for instance, octopuses, whilst they've failed the mirror mark test as a demonstration of self-recognition, they appear to recognise others, so specifically individual human caregivers. Mimic octopuses adjust their mimicry behaviour in response to different species in their environment. They will actually mimic different animals depending on who is watching them. Day octopuses hunt with a variety of different reef fish. Could this mean that they take on the perspective of their hunting partners? And finally, cuttlefish can change their appearance to mimic members of the opposite sex, which has been described as a form of tactical deception, an ability which suggests they take on the perception of others. So thank you. I'd just like to acknowledge some of the researchers that contributed to the studies I presented today and some of the funding bodies that have funded my research.
Thanks very much, Alex. We are now moving from the cephalopods to the decapod crustaceans, crabs, lobsters, shrimps. And to speak about these animals, we have probably the world's leading expert on them in relation to questions of sentience, Professor Robert Elwood, or Bob Elwood, who has been working on animal behavior for a very long time. He finished a PhD in paternal and maternal care in rodents in 1975. He became a lecturer at Queen's University Belfast in that year, and later a professor in animal behavior. His research on motivational change during crustacean contests provided a platform for studies on possible pain in that group, and he'll be telling us about some of those results. Bob Elwood.
Good afternoon. Thank you for that very generous introduction, unexpected and unwarranted. I'm going to talk today about pain, specifically pain rather than consciousness specifically, in decapods. And we first have to think of nociception. This involves receptors that respond to noxious stimuli. These stimuli then trigger reflex responses by which either all or part of the body is withdrawn from the stimulus. Now, this provides essential protection to the animal. Now, humans have an additional capacity which we call pain. Here, nociceptors also send information to the brain, and this generates an unpleasant feeling that we call pain. The question that arises is, if the nociceptive reflex stops the tissue damage, then what's the point of pain? And the answer normally given is that because the experience is unpleasant, the animal may change its future behavior to avoid that feeling. It enhances fitness by reducing future tissue damage, and it enhances recovery and repair of that tissue damage. So we're left with this problem of distinguishing between nociceptive reflexes and possible pain responses, and that's not always a trivial task. And I'm going to look at 5 markers that might help us in this task. I'll talk to these one at a time. First of all, avoidance learning. And the method we used in a straightforward, fairly simple experiment was to have an arena which was brightly lit, and there were 2 dark shelters, one at each end. And we placed shore crabs, which do not like bright light, in the center of the arena, and they reliably run to one or other of the shelters. And we selected crabs randomly to receive a shock or not in the first shelter they selected. If they received a shock in that, they wouldn't receive a shock in the others. The other crabs weren't shocked in their first choice, but they would have been shocked had they gone to their second choice in later trials. And we used repeated trials. We delivered shocks by attaching wires to their hind legs, wrapped around the flexible joint where there's no protection from the exoskeleton. So just getting to the results very briefly, these crabs showed very swift avoidance learning, and they quickly learned which shelter was safe. They changed their behavior after just 2 trials. During the experiment, some crabs left the shelter, but only if they were shocked. These data are what I typically refer to as are consistent with the idea of pain. I'm going to move now to a second experiment which looked at 2 criteria, prolonged directed rubbing, first of all, and here we used glass prawns. I don't know whether you can see here on these prawns. They have very long antennae, and it's the antennae which were the site of the noxious stimuli. So briefly, it was a 2-stage experiment. We selected one antenna at random, toss of a coin, and that was pre-treated, again randomly selected, with either water being brushed on half of that antenna, the distal half, or a local anesthetic. It was benzocaine. 5 minutes later, the prawn was taken out of the water. The same antenna was treated again, either with water, sodium hydroxide, or acetic acid. We noted that prawns showed 2 key activities in response to some of these treatments. If they were treated with chemicals, those noxious chemicals, they responded by grooming of the antenna, the specific antenna, not the other antenna, the specific antenna. They repeatedly pulled that antenna through the small claw on their first appendages. They also showed another behavior. They rubbed that antenna against the walls of the tank. Now, these activities were markedly reduced by prior local anesthetic treatment. And these sorts of experiments we've seen, these directed rubbing and grooming, we've seen in experiments on shore crabs where we've used chemicals, or hermit crabs where we've applied electric shock. So I'm going to look now at motivational trade-offs and long-term behavior changes in one experiment. Again, it's a 2-stage experiment. Hermit crabs on the shores of Northern Ireland, the small hermit crabs, occupied 2 main species of shell. There's Littorina shells, which they really like, and there's gibular shells, which they really do not like. Given any choice, they will take the Littorina, and as in the photo you can see here, they will initiate fights if they're in gibular shells and attempt to get the Littorina shell from the other crab. So we use this shell preference in part of the experimental design, and we induced crabs to occupy either a good quality, that is a Littorina, or a poor quality gibular shell. And these shells were wired up so that we could deliver electric shocks. The shock was delivered within the shell to the naked abdomen of the crab. Shocks were given to some crabs at a voltage just below that required for most crabs to exit the shell. Other crabs were not shocked. In fact, when we shocked the crabs, we really didn't want them to get out of the shell. That's why we were trying to keep the shock level quite low. But of course, some of them did get out of the shell, but only if they were shocked. No non-shocked crab got out of the shell. But we noted, and this was just a fortuitous result, significantly more moved from the poorer quality shell. This shows there's a motivational trade-off between avoiding the shock and keeping a valued shell. And this is clearly a behavioral decision rather than a nociceptive reflex to the shock. If it was just a reflex, it wouldn't matter what shell they were in, they would show the same reflex. Looking now at long-term change in behavior, which was in fact the intention of the experiment, after these treatments, crabs were offered an empty shell, and those offered shells following shock reacted markedly different to those that were not shocked. They were more likely to approach the new shell and more likely to move into the new shell. When crabs investigate empty shells, they feel the exterior of the shell, then turn it to access the aperture, then they repeatedly insert their chelicpeds, that is their claws, within the shell and sort of measure the shell and get information about the shell quality by those actions. Those crabs that were shocked markedly reduced this investigatory behavior compared to those that were not shocked. That is, they seem to show all the characteristics of devaluing the shell they were in if they'd been shocked within that shell. This shows that the subsequent change in behavior cannot be a reflex. It happens sometime after the shock had been delivered. Their motivation to keep that shell had been markedly changed by the shock, and that lasted for at least 24 hours. That was the longest we did between receiving a shock and being offered the shell. So this clearly cannot be a reflex. It is a long-term motivational change. So these data on crustaceans cannot be explained purely by nociceptive reflexes. We see rapid avoidance learning, and that's been shown in various studies. We see prolonged directed rubbing and grooming, and that's been shown in various studies. We see effects of local anesthetics, trade-offs, and that's been shown in a variety of cases with trade-offs between avoiding predator odor and avoiding shock, or avoiding bright light and avoiding shock. We see long-term motivational changes to noxious stimuli. We've done a range of experiments. We've seen other things, like brown crabs will guard wounds when confronted by a conspecific. If they've been injured, they will cover that wound with a chelicped. Shocked animals become very wary about the environment. They avoid risks, that is, they show signs of anxiety. Injury or electric shock also causes rapid physiological changes. That is, they show stress responses that are specific to those stimuli. And hermit crabs that leave their shells, or shore crabs that leave their shelters, are giving up valuable resources to avoid the stimulus. That is, they will pay to avoid those noxious stimuli. So these data are clearly consistent with criteria of pain, but that's not the same as proving pain. Nevertheless, it's the accumulation of evidence across different species that indicates we cannot dismiss the idea of sentience in these animals. We should give them the benefit of the doubt. Indeed, it's sort of heartwarming, although it wasn't my intention when I set out on this research program, it's heartwarming to see that that is in fact taking place. They are beginning to be given the benefit of doubt with changes in attitude by a range of organisations and a change in the legal status in Switzerland, and now due to the magnificent review by Jonathan and co-workers, very nice to see a hermit crab on the front cover of that. We see a change in the law regarding sentience of these animals within the United Kingdom. So thank you very much.
Thanks very much, Bob. And now we move from decapod crustaceans to insects. And again, we have the privilege of being joined by undoubtedly one of the world-leading experts on the taxon in question, Professor Lars Chitka. Lars is a professor of sensory and behavioral ecology at Queen Mary University in London and author of the book The Mind of a Bee. His research has established links between sensory physiology and learning psychology on the one hand and evolutionary ecology on the other, addressing questions such as why do animals have the sensory systems they do? How do they use them in their natural foraging environment? How do cognitive and behavioral processes function in the economy of nature? His work has made use of field studies as well as experimental studies with computer-controlled behavioral tests, computer simulations, and phylogenetic analysis. And in recent years, he's started to turn his attention to questions of sentience in these animals, and he'll be talking through some of the most striking evidence from that work. Over to you, Lars.
Okay, so it's very, very moving and exciting to be here. It's such an important conference. So what's the evidence for me to base headlines such as "Do insects feel joy and pain" on? So where I come from, I used to just study sensory processes. Oh, I need to stay. Oh, that's going to be hard. I'm a very Latin speaker, even though I don't look like it. But all right. Well, I'll try my best, but nudge me back when I stray too far. So what bees have to be good at naturally is they have to be careful shoppers in the flower supermarket. So in any bees' flight range, there might be several dozen flower species, all differing in color, scents, rewards, that they need to learn to pick the ones that offer the highest rewards for the least cost, and then learn the sensory signals that come with these rewards and focus their efforts on just that. And that is simple associative learning. It has nothing to do with consciousness or indeed thinking. But it's, in the first instance, remarkable just how flexible bees are in associating anything for which they get a sugar reward. What you see on the right here is a honeybee about to land in front of an image of a human face with a little drop of sugar water. And if they do that a few times, we can then give it the equivalent of a crime witness test where we present various different faces and ask it to identify the one that they have previously experienced a reward on. And they can do that just fine with about 80% of correct choices, even for similar faces. This was actually the only time that I had some interest from the US military in my research about 20 years ago. I gave him a short answer. But the flexibility, of course, is already impressive. But this here is a little bit more than just associative learning. What you're seeing here is a bee has been taught to roll a little ball from the periphery of this arena to a goal area in the center. And she can learn that just fine. So she rolls it backwards, and then she gets a little reward. And you saw another bee there that actually had no involvement with the ball rolling. And in fact, this bee has never rolled a ball for reward before. Number one is, if they observe a skilled bee just a few times, then the observer bee will manage to solve the task herself. She will then roll a ball that she's never done by trial and error. She will just get it that you have to roll that ball to the center for a reward. But moreover, the most remarkable thing is, of course, we've played a trick here. There are 3 balls, each at different distances from the target. Best way to solve this is to pick the closest ball, right? That's obviously the energetically best thing to do. But the experienced bee knows she can't do that because we've glued the 2 closest balls down. They can't be moved. So that's what the observer, the naive individual, gets to see. And we're then putting that individual on the spot and say, can you not just ape what the demonstrator did, but solve the task in a better way? And in that case, of course, we don't glue the balls down. The observer spontaneously, without trial and error, picks the closest ball, which indicates to us that there's some form of understanding of the desired end state, not a copying of the actions. What we're seeing here, we're now asking, do bees have mental representations of things? Now, bees are obviously good at recognizing patterns. We've heard that already. That in itself doesn't require a little virtual image, a mental representation of an object, because you can solve pattern recognition tasks with very simple feature detectors. What we're asking here is, can bees recognize an object in a different sensory modality? In the same way as the old birthday party kiddie game where you reach into a bag blindly and you identify a key, a spoon, a screwdriver just by touch when you've previously only seen it. The sensory input is completely different. You have to somehow be able to picture that thing to solve the task. This bee's been trained in complete darkness to find these shapes under infrared light by touch, and is then asked to discriminate cubes from balls using vision alone. And she's never seen these objects before. She's only felt them. And you can see she looks at the balls there, inspects them from all sides. She can't touch the shape now, but focuses her interest in the cube so that it can recognize the object in a completely different sensory modality. Now, none of this so far has anything to do with feeling stuff. But this question over decades of working with intelligence tests led me to wonder whether there might also be emotion-like states. If they're that smart, maybe they can also feel things. And I would have dismissed this, actually I did. People pointed out to me 20 years ago as a ludicrous question. But this is an experiment that changed my mind a little bit. We were interested here 15 years ago with Tommings about whether bees could learn about predation threat. So one of the things that bees have to look out for are these kinds of crab spiders that lurk on flowers. They are often hard to detect because, like a chameleon, they can adopt the color of the flower on which they prey. So they're hard to see. We took this situation into the laboratory with what we called a robotic crab spider, as you can see here on the right, where a plastic model of a spider was paired with two sponge-clad pincers, briefly capturing a bee, then releasing it, as in an unsuccessful predation attempt. And the bees learned this just fine. That perhaps is unsurprising. They learn to recognize the spiders, even if they have to do shape from shading, so if there's no color difference at all. But their whole demeanor changes, so they will then inspect every flower very carefully before landing, and only after several seconds of scanning land on the flowers. But the really surprising thing for us was that they displayed, even 24 hours after such predator attacks, simulated predator attacks, lots of false alarms. They'd inspect a flower that was perfectly safe, like with these scanning movements, and then reject it, as if they'd seen a ghost. And that, of course, is indicative of, well, it's the same sort of situation when you're walking through a dark forest. You might be anxious. You start seeing sort of shapes in the shadows under trees and so on that are not actually indicative of anything threatening, but because your attention is cued in that direction, there might be something scary, that's what you feel. Now, this isn't yet a formal test of any kind of sentience. It just nudged my mind in that direction. We confronted this question head on with the kind of experiment that we've heard about earlier, these kinds of motivational trade-off experiments where you're asking your animal, is your glass half full or is it half empty? You give it an ambiguous stimulus and ask, is that more likely something good or something bad? And we took that, adapted this for the bees world by training them first that one color, let's say blue, is always good, green is always bad, there's never a reward, and then confronted the bee with ambiguous stimuli, turquoise. And it turns out that the judgment of this 50% filled glass, the turquoise stimulus, depends on what happened to the bees before the experiment. If they had had a little surprise reward, they get a little dopamine hit, actually, so this is dopamine mediated, which induces them to view the ambiguous stimulus as more likely positive. And Jerry Wright at Oxford University did the opposite, where an ambiguous stimulus was presented after a simulated predator attack. And in that case, the response to the same stimulus is more likely, is met with reluctance to land on. Now, we've heard already about the crucial distinction between pain and nociception. Just responding to an unpleasant stimulus can be done with a reflex loop, and it is in many animals. It does not necessarily mean that you have an effective component to pain, a kind of ouch-like sensation. And that, of course, is important. Just showing that the animal responds to something unpleasant is not enough. But we've heard a number of examples just now already from Bob about so-called motivational trade-offs, where you have that noxious stimulus, but you prioritize something else that's precious. And so what Matilda did in these experiments, she's here in the audience, I believe, some over there, with a number of different experiments, but this is one of them, is she first gave bees 2 different feeding stations, shown on the bottom left there, one yellow, one purple, and they're equally rewarding, but the left one is hot. And not actually so hot as to be injurious, but hot enough to be, I don't want to land there. And they learned this clearly and prefer the purple unheated platform, as you can see with the behavioral results there. If, however, you make that heated platform more rewarding, then they essentially grit their teeth. This is the scenario on the right side here. So now we have 40% sucrose in the heated feeder. And then they go, well, okay, this hurts, but I want it. And so they go there and land on this platform. Even if you turn this into a memory experiment. So it's crucial, of course, for there to be some sort of mental process at work that you retest the animals under conditions where the stimuli isn't currently available. And even then, the bees prefer from memory what used to be the heated station. Now, we've done all these experiments with bees learning to recognize and roll balls, and thus led naturally to the question, I guess, for another PhD student, Salmadia Galpayaga, whether they actually like to play with balls, even if there isn't a reward. And it turned out that the answer is yes. So even in a situation where no reward was ever offered other than the activity itself, the bees returned again and again to this playground, as we call it, where they knew there's not going to be any sugar, ever. And indeed, the preference for that chamber increased over time for many individuals before they got a bit older and out of play age, and then the preference declined again. But it was principally an activity that young bees engaged in. Also, perhaps unsurprisingly, male bees did this a lot more than female bees, and that's because they're actually perfectly useless for any work in the colony, so they have a lot more time on their hands. So just to wrap this up, from these studies and others from my colleagues, it seems at least very likely that there are mental representations of space, time, and number in bees. There is tentative evidence that bees indeed can't just solve problems by thinking, in other words, not by trial and error and simple associative learning, but that they can also feel, at least using the same criteria that are used in other animals. There are positive and negative cognitive biases and play-like behavior. There are long-term memories from adverse experiences, false alarm responses indicating some sort of anxiety-like state, and at least all the evidence that we've collected so far, Tilda has collected for pain experience, also looks positive. And I suspect this is not limited to just bees. We use them because they're cooperative with such experiments, but very likely also to the many other insect species that are farmed in industrial processes that have at least the potential to be very unpleasant in terms of their slaughtering methods. And I think, to me, this instills a new kind of respect for the natural world. I think that with this, everything we've heard today, we're really witnessing a kind of Copernican-like revolution. We are not alone as animals with minds. We are essentially enjoying, feeling, suffering, thinking beings in a world of other enjoying, thinking, suffering, and feeling beings, however different their priorities and perceptual worlds might be. But there are lots more out there, I'm sure, that we will discover in future years. Thank you very much.
So I could invite all 3 of our panelists in this session to take their seats at the front.
All right.
And then we'll start things off with a question that is for all 3 of you. And that is simply, can you tell us about the single experiment that did the most to shift the way you think about these animals? Should we start with Bob?
That's a difficult one. Which is your favorite child? It's that sort of question. And I think, though, I would go back to the very first experiment that was published, and it was the one, God, what was it? It was the one with the glass prawns and the local anaesthetic. And I have to give credit to Victoria Braithwaite and Lynn Snedden, who did an experiment on trout and injected acetic acid and gave them morphine. And if they got acetic acid injected into the lip, they rubbed the lip against the substrate of the tank. And if they received morphine, they didn't see that behavior. And that was a guide for me for the first step into crustacean research and the possibility of pain. Couldn't use morphine at the time. It takes an awful lot of time to get a license to have morphine in Northern Ireland, and a lot of begging and pleading and strange questions. But the approach on that experiment went beyond the fish lip experiment, in that we were using a several-stage process and looking at multiple behaviours at each stage. And the analyses did, in fact, get quite complex. But I think it was the thoroughness of that was borne out by the impact it had, which took me completely by surprise when journalists started to phone up about an experiment on prawns. And we were bamboozled by a half-page article in the Daily Mail, wish it had been a decent newspaper, but a half-page article on the experiment. But the next day, there was a 2-page spread on that experiment in the Daily Mail. It was just absolute nonsense. So that had the impact and the impetus for us to carry on.
Great. And Alex?
Oh, I'm finding it hard to narrow down. So I'm just going to talk a little bit about my research at Cambridge, where I showed that with my colleagues that cuttlefish can remember the past, exert self-control and plan for an imminent future. And when you say it like that, you might not think about how complex these traits are. So I want you to consider that you can remember which restaurant serves your favourite meal, where it's located and what time it opens. Now, in a similar manner, cuttlefish can remember where they ate their last meal, what their last meal was, and how long ago. Now, imagine you're sitting at the restaurant and they serve this irresistible freshly baked bread, but there's a catch. Sometimes if you eat the bread, the waiter won't bring your main dish. And so knowing this, you exert self-control and you don't eat the bread so that you get the lobster. Now, cuttlefish can delay gratification to get superior rewards in this similar way. And now consider that the restaurant only serves your favourite meal every second night, and it's a really big portion. So in anticipation of this, you skip lunch that day to turn up with ample appetite, and cuttlefish also can do a similar thing. So they have incredible foresight to anticipate what meals might be on the horizon. So they'll eat less crab for lunch when they can predict that shrimp is on the dinner menu. And these sorts of traits, in humans at least, have been linked to consciousness, especially the recollection part, because humans have an idea that that memory belongs to them, and also that it occurred in the past. Now, we can't say the same thing yet for animals because we don't have any agreed behavioural markers of consciousness, but the behavioural evidence that we are seeing is very compelling, and it's hard to say that these traits uniquely emerged in humans. These traits have an evolutionary history, and we're seeing very large similarities between these traits even in animals that diverged from the human lineage over 550 million years ago.
Could you briefly explain the experiment you did that was inspired by the marshmallow test?
Yeah, absolutely. So I adapted the Stanford marshmallow test, which was conducted in the early 1970s, to test whether children had self-control. And so what would happen here is a researcher would give a child a marshmallow and explain to them, if you don't eat this marshmallow straight away and wait 15 minutes, you get a second marshmallow. And this was really hard for some children, and they would do all these sorts of behaviours to try and distract themselves, like they'd look away, They'd try and put their fists in their mouths. They'd try and distract themselves. And it varied. Self-control can vary a lot between individuals. So since that test, a lot of comparative psychologists have adapted it to test for self-control in non-human animals. And I adapted it to test for this in cuttlefish. Obviously, you can't tell a cuttlefish to wait. So what I did is I had these two different clear chambers with a preferred food and a less preferred food in the chambers, and they learnt to associate that the chamber with the less preferred food, which they still readily ate, would open immediately, and the chamber with the preferred food would open only after a delay. And then when they were given both chambers at the same time, they had a decision. They had to either suppress the temptation for the immediately available food, or they could eat it straight away, but that meant that the preferred food in the other chamber would be taken away. And so I think at the time, I mean, they always just they surprise me. I think I was expecting for an animal that lives 1 to 2 years to see maybe self-control in a matter of seconds, and that's what we see in rats and pigeons. But we saw an ability to delay gratification for up to 2.5 minutes, which puts them in a range of what we see in crows and chimpanzees. And so that really blew my mind. And like in children, we saw a difference across individuals as well. So some were better than others when they were exerting self-control. Some of them even looked away from the tempting food in front of them as if they were trying to distract themselves. And the individuals that actually did better at self-control or waited longer for the delayed but better reward also did better in another cognitive test, which is the first evidence of linking delayed gratification and general intelligence in an animal outside the primate lineage.
And Lars, what about you?
I'm going to decline to pick a single experiment because I don't think there is one. So I think my best argument in favor of there being a mind in bees is all these experiments together. And this is because, of course, for every individual experiment with the bees or other animals that we've heard today, someone with wielding Occam's razor too wildly will say, well, there is an alternative simple explanation. But the point is, for all these markers of consciousness, let's say in bees, we haven't yet found a single negative experiment result. And while for each individual experiment you might find some sort of alternative explanation, you don't have a simple explanation that fits all of them. And so I think the arguments that where people try to invent what I feel are increasingly more convoluted explanations to invoke associative processes for all of these phenomena are really equivalent to flat earth people trying to ever more complicated, convoluted explanations why the earth might after all be flat. You could use, you sort of look out of the window and it looks that way at first glance, but the moment you probe a little deeper and want to find an explanation that fits all of what we've heard today, I think we can't get around the idea that at least in most of these animal species that have been looked at today, there is a conscious mind behind them.
Okay, I'd love to open this out now to questions from the audience in the room and also online. I'll start with one from the online audience, which is a question for Lars from Katerina Bukova. Could the play you observed in the play study be simply exploration?
Good question. We wondered about that because, of course, naive bees that have never seen flowers before are by default interested in colored objects, even before having ever made any experience with them, and will handle them and try to find a reward in them. However, when the bee thinks it has a flower in front of it, it very quickly gives up if the flower is rewardless. And so there might be 2-3 trials, no reward, the bees will abandon it. So in this particular case, the repeated returning, where often they roll these balls about for minutes and minutes, happened with a completely different dynamic. That is, they wouldn't try and then abandon, but actually the preference for the area with the movable balls increased over time with experience. And of course, the bees had been given food ad libitum, so they had as much food elsewhere as they wanted, yet they came back again and again, making detours, paying a cost to get back to these balls, which seems to indicate that really they were not looking for sugar rewards there, at least not after the very first trial, perhaps.
Great. Let's take one from the room now. Yes, let's go to the end of the row there. Sophia, yes.
Oh, hi. Hi. Can you hear me? Yeah.
Yep.
So this is a general question to any of the speakers. So we've been hearing a lot today about the negative marker of pain, right? Electric shocks, noxious stimuli, wound guarding, and so on. And even some of the psychological responses such as anxiety and stress which might result from those kinds of experiments. And so my question really is, why can't we get beyond pain as a marker and move to more humane kinds of experiments which may, such as pleasure, seeking pleasure, which is also a marker for consciousness. Wouldn't the presence of pleasure seeking be the same, yield the same kind of results in terms of what we're looking for in defining consciousness?
Yeah, so I think, you're absolutely right. When you look at the field of animal sentience, there's a huge skew towards the investigation of negative emotions. And that's because they have the most implications for ethics and welfare. And people in this field are... Well, what these researchers are trying to do is come up with practices and policies to reduce and minimise those risks to animals. Now, the field, or the researchers in the field, are aware that there is less focus on positive emotions, but I think the field is changing. And it is really important to take a look at the other side of sentience, that positive emotion side, because without understanding that other side, one, we don't get an understanding of the evolution of sentience. We're only looking at one half of the coin. And two, you're right, we don't really, like, in order to create holistic practices and welfare for an animal, we can't just take away anything that might cause harm, because that's completely underappreciating their capacity to feel positive emotions and live a well-rounded, enriched life. So it is really important that we turn focus onto those potential positive emotions that they may feel, because placing them in a room that is free from anything harmful isn't the same as placing them in a room that, yes, doesn't have harm, but also provides them with an environment that evokes positive emotions. And so I think researchers in the field are aware of it, and we are moving towards it. It's just probably taking longer than we'd hoped.
Bob?
When I started to look at this, it was pain that I was interested in. And I think that pain is more important than pleasure with regards to crustaceans, because there are billions of crustaceans used in the human food chain each year, and they are subjected to being butchered prior to being killed. They are boiled live. They have their main claws ripped off, and then they're thrown back into the sea alive. And it's important to know whether these things might be harmful to the welfare of the animal, to how it feels. And I felt that that was a stronger motivation for research than to ponder whether a hermit crab feels pleasure when it gets a preferred shell.
Lars?
I mean, I take your point, and we do, in fact, look at a diversity of emotion-like states, as you've just heard. But The point, I think, is, and I think you two already hinted at that, we are surrounded by a planet where the vast majority of the public and policy makers, specifically in the case of invertebrates, would hold that there is nothing to worry about whatever you do to them. That's what the vast majority of people out there believes. And we can't really change that viewpoint without confronting this uncomfortable issue about whether they are capable of suffering. So there are millions, billions of insects used for invasive neuroscientific treatments every year in labs around the world, and I don't even want to talk about the insect feed and food industry. But they operate in a welfare-free space because the historical wisdom is there is no welfare to consider. They don't feel anything. And they wouldn't be convinced, I think, by, let's say, a single study on insect play. We need to, however uncomfortable that is, at least use some moderately unpleasant stimuli to determine with some certainty, is that unpleasant for the animal? Otherwise, we will not convince anyone, in my worry.
Yep, so we've got some great questions coming in from the online audience. There's one here from Luke Rolofs. The research on bees all seems to concern adult insects, but much insect farming involves slaughtering them before this as larvae, which are anatomically and behaviorally different. What can we conclude about larvae from experiments on adults in metamorphosing taxa? Lars?
Good question. We can conclude nothing about larvae from the work that we did on adult bees. And so this is a huge evidence gap. There is a bit of work on learning behavior in fly larvae, but nothing to address specifically the question of sentience. Watch this space. We are actually in the progress of launching exactly this sort of work with Jonathan and some young scientists who've just started working on this in our lab because it's a very important issue.
Let's have another one from in the room now. So right by where you're standing, Sophia, let's have the question from there.
Hi, I'm wondering if you have any comments on other types of mollusks, I guess particularly gastropods, and if there's any research being done, I guess on especially like more prolific species, thinking like land snails, aquatic snails, slugs. Thank you.
No, sorry, I don't know. There might be out there, but I haven't looked into it.
Okay, so let's go to another question online. This is from Georgia Mason, who writes, fascinating stuff, but please could someone explain what the evidence is that motivational trade-offs require conscious feelings as opposed to simply representing the interaction between an implicit approach rule and an implicit avoid rule?
When I set out to do some motivational trade-off experiments, I wasn't thinking that they would provide evidence of consciousness or any greater ability. What I was looking for was evidence whether it was a reflex or not. And it clearly isn't a simple reflex because what the animal does in response to the noxious stimulus depends on other motivational states. So it is showing that it's a behavioral trade-off. Sorry, it's a behavioral decision. And that was the key to me. I was asking, is it a reflex or is it a bit more than a reflex? I never wanted to go over to saying it was going to be much more than that.
Lars, do you want to talk about the B version?
Yeah, so I think the question is well justified because you could, even in very simple organisms, even ones without a nervous system, they respond with attraction and rejection to certain chemical stimuli, even in single-celled organisms, so long as these stimuli are currently present.
Yeah.
So a key question, and this is of course at the core of these motivational trade-offs since they were first brought up in the question of insect consciousness by Auguste Forel in about 1901, is whether you're trading off things that are currently out of sight. So he discussed the case of ants trading off a threat versus a distant reward, but they were both in memory rather than currently present. And that, of course, is the key question. Whether it's, so you have to go beyond the trading off or the differential response to currently conflicting stimuli. But it's not on its own, of course, direct evidence for consciousness, but it's certainly evidence that there's something more going on than a simple reflex-like behavior, as Rob said.
I think that's really crucial, that there's trade-offs of different kinds, some more sophisticated than others. And a feature of both your work with Tilda, Lars, and your work, Bob, is looking for trade-offs of a reasonably sophisticated kind that is about how the animal represents value and disvalue rather than just about the immediate stimuli. And in Bob's experiments, this is about the shell quality, where it really seems like they need a representation of how good the shell is that they're in. And in Tilda's versions of the experiment, it's about their memories of experiences associated with the different heat pads. And in both cases, it seems like some evidence, at least, of a kind of, might put it as sort of inner map of the evaluative landscape, as it were. Remembering where the disvalue and value is and somehow weighing it in a common currency. At least that seems to be the argument. So let's have another one from within the room. I think it would be nice to go to this side, because this side's been relatively neglected. Thank you.
Hello. I have another particularly mollusk-relevant question. So sort of well-known, sort of confusing edge case in animal sentience is bivalves, particularly basic bivalves. Mm-hmm. In this context, one of the most troubling arguments I've run into, especially appealing to what other mollusks look like, is that some bivalves, it's possible that they have consciousness as a vestigial structure. I was wondering if any of you could comment on the possibility of retaining consciousness as a vestigial structure and whether that will ever be testable through any tests like these, or I don't know, just assuage my worries.
I didn't quite catch the question, I'm afraid.
Well, there's
so
the
if
question
someone
of,
else did, then
bivalves. Any thoughts on bivalves? They're mollusks, Alex. So you can be our mollusk person, I'm afraid.
Oh, I think this is out of my expertise. I can't comment. Do you have any?
Yeah, the idea of pain is that it may change the animal's behavior, not just immediately, but in the future. And for that, you need flexibility of behavior. There has to be choice available, and the animal has to be able to change their choices in the future. And I've sometimes wondered where you have animals which sit around that they're predominantly sessile, maybe attached to a rock. Either they open their shell or they close their shell. And I sort of wonder whether you need a pain system to be beneficial, whether it would be beneficial. And if it's not beneficial, I would expect it to disappear. I presume that having evolved this system, that it imposes a cost. It doesn't come free. And it would be quite pointless to generate with each new individual a pain system if it's of no use. And I would expect rapid selection against it.
So in scallops, there is this escape behavior they do, where they will swim away from starfish. So there is a limited behavioral repertoire, but it does look a great deal like a pretty straightforward reflex.
I was thinking more of an oyster, which is stuck to a rock.
And some display even less behavior than scallops, that's right. I did, my team at one point did review the evidence that exists for bivalves. It was mostly Heather Browning doing that work. And to be honest, there was very, very little. So to me, there is a gap. There's a very large gap between the quality and volume of evidence we have for the cephalopods and the volume and quality of evidence for bivalves. I want to take another question from the online audience. This is from Noah Weiss, who asks, I'm just curious as to the impact on animal research that you expect to have with your declaration. Some universities have a policy that they kill the animals they do research on when they're done with them. Do you expect to change this? I guess, for example, can you elaborate? I think it's fair to say the signatories will have a wide range of views on that one, but I'd be interested to hear your views.
So I think in insects, it's likely that some sort of change will implement it, because we're currently in a setting relatively unfettered by not just any ethical considerations, because very few people take them seriously, but also there is no law to protect the welfare of insects in research, for example. And given what the evidence that we've heard today, and of course from many other labs, it looks at least reasonable that we should take the possibility more seriously that there is that some of the laboratory procedures that are applied by my colleagues are quite profoundly pleasant. So, I mean, to give you an example, it sort of mirrors what Rob said about lobsters in restaurants. So many of my colleagues who do electrophysiology on insects will remove all their extremities, will embed what remains of the body in hot wax, and then insert electrodes into their nervous system. All of this without anesthesia. And of course, a successful experiment is one where the animal stays alive, ideally for several days in that state. And this is all based on the sort of convenient assumption, well, there is nothing to worry about. And so I think we can't uphold that concern-free environment anymore. And so I think eventually the law on these things is likely to change, which will make me unpopular with my colleagues, presumably. But I think it's fair that at least some consideration is given to in reasonable ways, where that is possible, to try to minimize the potential for suffering, to justify the severity of the treatment in proportion to the knowledge gain, to consider, as the vertebrate people do, numbers of individuals sacrificed for any one particular project, and so on. None of this is currently regulated at all, and I think that is a worrying situation.
Yeah, so we wrote a paper called "Is it time for researchers to consider for insect researchers to consider their subjects' welfare?" And we argued that the answer is yes. And we've both been involved with the Insect Welfare Research Society that is trying to enrich the evidence base with which we address questions like these. Any thoughts from Alex or Bob on this one?
Even though a lot of countries still don't offer protection for cephalopods, a lot of universities and different states universities have their own policies for working with cephalopods in labs, and we have to follow the same kind of ethical applications that researchers follow that work on vertebrates. So we reduce numbers. A lot of my work is non-invasive. We try and do lots of different behavioural experiments on the animals, and at least the labs that I've worked in on cephalopods, the animals die of senescence because, cephs only live for 1 to 2 years, so we don't have to kill them off at the end. I think in terms of more pain research, like in Robin Crook's lab, she also has to adhere to really strict practices and policies because the US doesn't protect cephalopods under animal welfare law, but different universities still require you to fill out those ethical applications. So she has to justify all the work that she does. And exactly what Lars said, it's kind of this weighing up of what can we gain from the knowledge of these experiments, how is it going to influence practices and policies and maximise the animal's welfare, from how much damage are we able to do to the animal.
In the United Kingdom, the law has changed in recognising decapods and cephalopods as being sentient. But with respect to decapods, there have been no legal changes in the United Kingdom about how those animals should be used. So in science research, they have pretty much the same status as a lettuce in what you can do. What I would hope is the immediate result of the research that we've done, is that people will not wait for legal change, but to regulate themselves in a sensible way, to apply the 3 Rs, try and keep the number of subjects low, try and keep the treatments moderate. But I think my greater hope is that rather than worrying entirely about a few hundred animals that are used each year in scientific research, we should wonder about the billions of animals that are used in the food industry, where there's no regulation, but hopefully consumers will start to push for change and more humane treatment of the animals they're going to consume. But that is where the maximum good would be achieved, I believe.
Okay, let's have another one from in the room. Let's come forward to the second row from the front. Please do wait for the microphone to come to you. Yes, thank you.
Hi, thank you very much for fascinating event, your presentations. I even was on Zoom in the morning and I decided to come in. But audio actually was better on Zoom. On computer than here. But it's, I understand, first I'm not from academia, I am outsider, I'm like general public. And it's understandable why so much attention to the pain markers, because it's most obvious, seems like. But in all these discussions, I never heard that even in humans, there are some conditions, some disorders when they don't feel pain. For example, probably people remember Sandy Hook mass shooting. The boy did not feel the pain who did it. So, but it's not like a response in England. There is a woman who also has this condition, but she's extra careful because she knows. So connection, definitely they are conscious. So this connection between pain and consciousness probably not that direct. It's first question. Second, that, okay, we all always do everything with our best intentions. But some people, after your research, using your research, could say, okay, let's genetically modify chickens. They don't feel anything, don't feel pain, and don't care how they raised, how they consumed, or any type of animal that we kind of could genetically modify for our for different reasons. So, and your research definitely could be used. So how we develop these ethical systems that prevent something that we even deprive all animals because we want to feel good about ourselves, we deprive them of feeling pain or any other feelings. Thanks.
So I guess these are two questions. I think the first one is, yes, you're right. One of the hallmarks of pain in humans is just how dramatically disjunct the perception of pain is from an injury. So there's a famous story where a construction worker jumped accidentally on a nail, which was so long that it actually went all the way through his shoe and out at the top. And the guy was, of course, brought to the hospital and was in such distress, and whenever someone moved the nail, was in such pain that they had to sedate him to remove the shoe, and then found that the nail had gone between the toes. And likewise, there's another mirror story where someone had accidentally shot a nail into his skull so deeply that the head wasn't visible, and thought that he went to the doctor 3 days later complaining of a toothache, but not being aware that there was a nail this size sitting in his head. And so this is why we're looking at the question of whether there is something more going on than a reflex-like response. Everything in humans about the link between, let's say, the severity and injury, which is mirrored in nociception, and the ouch-like pain, the affective component, involves plasticity and flexibility in responding to these stimuli. And that's why these responses, like motivational trade-offs, are so important, because they tell us that there is some modulation of the response where you can suppress the input depending on what the priorities are. That's why people are asking questions about endogenous pain modulation systems, such as invertebrates, the endogenous opioid system, and so on. That's the first half. The other is, of course, yes, you're right, there is the concern that that one nasty human response to the awareness of sentience of animals is to try to switch the sentience off. I think vegetarianism, veganism is a better response, or if you're that keen on animal protein, then of course there are now methods to generate that in a dish where there clearly are no ethical concerns either, which might actually be easier than to try to remove the centers of a brain that mediate the sentience or the pain perception.
Okay, I will take a question from online. This is a question from Nikki Clayton, who is one of our signatories, and I think we will hear from a video message from later. But Nikki asks, how do you interpret your findings within the framework of convergent evolution despite divergent minds and bodies?
I think that we, as comparative psychologists, are looking for similarities and differences when we look for different cognitive traits across diverse animal minds. Even though I think there was a tendency in the past to say that humans have unique traits, we're now seeing similarities of those traits in really diverse animal minds. And we're not saying that they work exactly the same or that the animal was having the exact same experience. There probably are differences. But I think it is really important to look at really different minds. It gives us a more complete evolutionary picture of how intelligence evolved or how consciousness evolved. And as Nikki also likes to point out, that if you're a computer scientist and you want to figure out how a computer is coming up with particular solutions to problems, you're not going to just study one particular type of hardware. You're going to look at a Macintosh and a PC and see how these different neuroarchitectures are coming up with similar or different solutions to the same problems that they're experiencing. And I feel like that's what we do in comparative psych. And even though traditionally we had a very narrow kind of range of what we were looking at, that range is starting to broaden, and invertebrates are coming into the conversation, and that is such an important part of the puzzle. It's going to give us answers to how intelligence evolved, why and when.
It's worth emphasizing, isn't it, that in these animals we've been talking about in this session, the organization of the brain is profoundly different from the brains of vertebrates. So it's a big difference with the session this morning. And so there's a sense in which we are definitely seeing examples of convergent evolution, just like with the eyes. You know, the eyes of cephalopods have evolved independently of our own eyes and show this weird mix of similarities and differences. Must be the same with the conscious experiences as well.
Absolutely. The cephalopod brain is donut-shaped. It's wrapped around their throat, and in the case of the octopus, they have 2/3 of their neurons in their arms, and they don't have a hippocampus, which is the memory and learning center in mammals. Instead, they have a vertical lobe, which has been shown to behave or function in similar ways.
Nice.
I think, yeah, in many cases, many aspects of consciousness that we've discussed today, and also they've been discussed in the literature out there, such as, let's say, mental perspective taking, metacognition, and so on, where they do occur in, let's say, primates and birds, they're almost certainly the result of convergent evolution. but it is at least useful to consider that some aspects of consciousness might be evolutionary ancient. And so Peter Godfrey Smith, for example, makes this useful point that disentangling sensory input that arises as a result of your own intended movements versus the same sort of input that comes from the outside world, disentangling that already becomes relevant when you are a mobile animal. So when you're tilting your head 45 degrees to one side, then your image shifts by 45 degrees. Nothing to worry about, you've tilted your head, that's why that happens. If you haven't intentionally tilted your head and the building is suddenly at 45 degrees, run for the exit because we're in an earthquake. But it's the same sensory stimulus. And he and others make this point that once you're starting to move as an animal, even with just touch receptors, you move your own worm head on an ocean seafloor, you bump into something. If you have initiated that movement, if that was your intention, and you bump into a rock, okay, that's a completely different stimulus to when you're sitting still, and it's other outside world generated input, in which case that might be a predator. So at that level, you already have a very important distinction of self from other, which might be, at least at that simple level, something that led to consciousness-like phenomena.
Let's take one more question from the audience in the room. I'll take another question from second row here. Apologies to those who had questions we've not been able to get to.
Hi, I'm Sarah. I'm interested what your thoughts are on the new emergence of insect farming, or really any, I guess they could be small aquatic species as well, in the name of climate change, just because we know very little about the climate impacts of insects and also about their sentience. Is this something that worries you?
It's a good question. And so there is an exponentially growing sector to farm insects. They do indeed greenwash themselves by saying that we are generating the same amount of protein as the chicken farmers, cattle farmers, and so on, but with much less greenhouse gas output, with less area coverage for the vegetation that needs to be fed to these animals, so all great. If indeed replacing conventional livestock was part of their goal, it isn't. Much of the insects, 99.99999% of the farmed insects, are used for feeding to chickens, to feeding to farmed salmons and pigs and so on. In other words, this is a mutually enhancing industry. It's basically the insect industry is turbocharging conventional livestock farming rather than replacing it. And the other thing that you often find on these companies' web pages is, in addition to there being less greenhouse gas emissions, the other good thing we do is there's no ethical concerns. Maybe that's the case for some insect larvae, but I think we need to see the evidence. We don't have it yet, and everything we know so far nudges probability in the opposite direction. And so I don't feel that I want to subscribe to the idea that making more and more of these insect factories without first evaluating the ethical evidence is a good idea.
Okay, it's been a real privilege to have three fantastic panelists and also two brilliant audiences, the audience in the room and the audience online. Thanks so much to everyone watching for all of your excellent questions. We'll be taking a 20-minute break, and then we'll be back to think about what the emerging science of animal consciousness means for ethics and for policy. So let's thank our speakers.